Okay. All right. Thank you very much. So my name is Meng. I'm a master's student at the School of Biomedical Informatics, University of Texas. And I am a group member of Dr. Tao's. And my background is in pharmaceutical sciences, and I have also worked a little bit in clinical research. So this project is a personal interest of mine as well. So the outline of today's talk will be followed. I will first talk about informed consent and its relation to drug development and why we care about donor permissions. Then I'll present my methods and results and propose some future studies. So what is informed consent? The purpose of informed consent is to assist a prospective subject in understanding the reasons why one might or might not participate in the research. And subject comprehension is the key to this process. Information should be presented in a way that a reasonable person understands what they're consenting to. And this emphasis is quite important, as we'll see later. It is not as simple as it sounds or as simple as you may think. And relevant to our study is this requirement, informed consent documents should contain a statement that after de-identification, the information or bio-specimens could be used for future research studies or distributed to another investigator for future studies without additional informed consent from the subject, or a statement that the subject's information or bio-specimens collected as part of the research, even if identifiers are removed, will not be used for distributed for future research studies. And as we all know, there are 4 phases of clinical trials, and informed consent process is involved in all the phases, even after FDA approval. And this picture is taken from Colorado Center for Personalized Medicine. It nicely illustrates the biobanking process for precision medicine. Again, informed consent process is always the first step. So why do we care about donor permissions? First, there is a growing interest in and rising value of bio-specimens and associated data. Stemming from the increasing need to build large cohorts, including combining specimens and data from multiple research or clinical resources collected in different environments and at different times. A current barrier to broad biospecimen and data sharing is the challenge of coordinating across complex sets of requirements that govern the collection and distribution of specimens and data. And in the United States, such collection and use for research is in part governed by regulations defined in 45 CRF 46, also known as the Common Rule. And remember that biospecimens and data can be collected under different environments. That includes under different regulatory environments, and regulations may change over time. So for example, a recent revision of the Common Rule requires broad consent forms for secondary unspecified future research, even if the biospecimen will be de-identified. And traditionally, biospecimens and data use and sharing depends on the use of paper-based forms for capturing participant consents, and then manually entered into a spreadsheet or database managed by the biorepository. And even with recent efforts to move this electronically, with electronic consent approaches, it will still not be possible to easily discover and then determine the status of permissions and constraints on sharing specimens or data without some sort of computationally scalable way of querying for information about those permissions and constraints. So how does our study relate to ontology? Due to the challenges to broad biospecimen and data sharing, there is a growing consensus that we need a complete ontology to model the common rule. And there are a few standalone ontologies for various types of clinical research, but integrated framework is still in its early development stages. This study was done as part of an effort with our collaborators to develop a more complete ontology model derived from the common rule. And with an automated method to extract donor permissions combined with our common rule ontology and informed consent ontology, we hope to help future research on permission identification. So moving on to our method, we first had to determine what permissions to extract. And after some research, we determined these 9 questions are most pertinent to donor biospecimen and data sharing. They broadly cover 4 categories: permissions related to sharing, permissions related to genetic research, permissions related to future research for commercial purposes, and also if donor recontacts are permitted. And here's a screenshot of our annotation process. We use a software called CLAMP, which is a clinical natural language processing software. Basically, we read through the informed consent documents, and if we determine a sentence that it contains an answer to one of the 9 permission questions, we will mark that sentence. So for example, in this document, here's a sentence that we marked as donor data permitted to be used for future research, and also the next sentence. And here is a sentence marked as biospecimens permitted to be used for commercial purposes. And the next one is the same. And our, I'm just pointing out some important points in our annotation guideline. This is not the full guideline. So for one, we only mark up text that clearly states what will happen to the donor, to the person specifically, or to you, and not a description about the study itself. And another important guideline is we only consider sharing as sharing outwards of the immediate study team. So the followings are not considered sharing. For example, sending sample to lab, or summary reports in journals or publications released for mandatory regulatory policy reasons, such as to the FDA, NIH, or IRB, or people who have access to database or data bank for operational reasons. And sending research finding to a medical record is also not considered sharing. And as you can probably imagine, this opens for a lot of interpretation, and it will be challenging to annotate those informed consent forms. So after drafting our initial guideline, we proceeded to develop our gold standard. And our corpus included 120 informed documents collected from online resources, including websites of IRBs, clinical and translational research groups, Office of Sponsored Program, Office of Research within universities, hospitals, research institute or biobanks, etc. And first, 2 annotators annotated the same documents for a total of 5 rounds. And after each round, kappa scores were calculated to evaluate the agreement between the 2 annotators. And we will discuss and update the guideline. And after 5 rounds, the 2 annotators would split the remaining documents and annotate them independently. So I am one of the annotators, and another one is a scientist in our group. And the next step is to take the annotated files from CLAN as showed before, and transform the files into a parsable format. In this case, we transformed it into a CVS format. And then all sentences were tokenized to remove any stop words, etc. And then converted into a term frequency and reverse document frequency features using Python's scikit-learn library. And we approached the problem of predicting whether a sentence in an informed consent document contains answers to the permission questions described as a supervised text classification text. And because of the dominance of blank sentences, as you can imagine, an informed consent document only has a few sentences that actually describe those descriptions. We broke down the classification task into two subtasks. The first one is classification of relevance, defined as either being blank or not. And the second step is classification of permission type as defined as any of the 9 permission questions. And we tested 4 traditional machine learning methods, including random forest and 3 different kernels of support vector machines. And this data set was, as I said, highly unbalanced. Only a few sentences in the document contains answers to those permission questions. And as you can see here, the ratio of blank versus relevant is quite high. It's actually above 20-fold. And so we had to employ some oversampling techniques. First, we analyzed the distribution of the number of words in blank and relevant sentences, and found that more blank sentences had shorter, more blank sentences have less than 5 words. So we use this as a basis to balance our data set. We cropped our data set to all sentences that contains 5 or more words. And then we use an algorithm called synthetic minority oversampling technique, or SMOTE, to oversample the relevant class in both the complete and cropped data set. And this table summarizes the kappa scores for each of the 5 rounds of synchronous annotations. And as you can see, although the kappa scores tend to increase over the 5 rounds of annotations, overall they are quite low, meaning the agreement between the 2 annotators are quite low. And among the permission questions, Q8 and Q9, which concerns the commercial use of donor data and recontact, have the highest kappa scores, and these are relatively easy to annotate. The document usually clearly says, we will, do you give permission for us to recontact you? But the others are quite difficult to annotate. And Q4, which is data permitted to be used for genetic research, was the least agreeable. And this particular question was difficult, partly because it involves donor data, which covers a wide range of information, such as patient medical records, genetic data, study results, or any unspecified data mentioned in the document. And also, this was problematic because it was hard to determine if the sentence directly describes permission to use genetic data or describes the study. because a lot of studies involving genetic research will spend a lot of the content describing how they will, what is genetic study, what they will do, and this is hard to distinguish between donor permission. And the results of our first step, which is relevance prediction, is summarized here. The models generally perform better on the crop data set compared to the complete data set. And the linear SVM model highlighted here had a comparable performance to the best model. Sorry, but was much faster to train. And since the next step requires repeated binary classification, we chose this model to predict the specific permission types. And the table summarized, so this table summarizes the linear SVM model performance split into relevance yes or no. And due to the still imbalanced data set, even after oversampling, the model performs better on predicting blank versus predicting relevant sentences. And the overall accuracy of our model was quite high. As seen here, they're all over 90%, but the model performed well mostly on classifying whether a sentence is not a specific permission question, or the label no here, compared to compared to the yes labels. And among the permission questions, our model performed worse on Q6. Donor data permitted to be used for future research, and compared to Q5, which is a similar question, it is bio-specimen permitted to be used for future research. And this also could be due to the challenge in identifying donor data, as I described before. So, what strikes us is the overall low CAPA scores, and this demonstrates the striking difficulty discerning the expression of permissions in informed consent forms. And also, this is due to the requirements in Sankan forms itself. This is not a priority. The priority is to describe the study, the risks and benefits, and etc. So the ambiguity in the informed consent language translates into the difficulty to build a high performance natural language processing model to annotate automatically donor permissions. And coarse granularity in the documents simply means it lacks the detail to give us such information. And to the best of our knowledge, this study is the first attempt to annotate automatically informed consent documents using natural language processing methods. And even though much room exists for improvement of the machine learning models, by using a simple and fast algorithm such as a linear support vector machine and achieving the results that I presented here, this study illustrates the possibility for building tools to promote biorepository data regulation, sharing, and possibly decision support. And the limitations of our study are obvious. We use simple machine learning methods, and we only have a relatively small data set to train our models. So a few suggested future study would be to prove model performance, possibly using deep learning methods, and using a larger corpus, maybe have an external validation set, and how this study can improve informed consent ontology usage. So I would like to thank my advisor, Dr. Tao, for the guidance in this work and all her help in writing this manuscript. And I would also like to thank our group members, Madhuri, who's the other annotator, and she developed the initial guideline, and Dr. Du and Dr. Xiang from our group, and Cooper and An from our collaborators from the University of Michigan. And thank you very much.
Great. So it's a very nice talk. Let's see, do we have any questions?
I have one question.
Okay.
So your data is quite unbalanced, and that's why you use the SMART. I wonder whether you examine the effect of SMART, leading to overly improved performance. Have you also tried something like more downsampling or kind of balanced sampling?
Yes, the results are not presented here, but yes, we have tested whether using SMART to actually improve the model performance, and it actually does. And also, we also tested a one-step prediction, meaning just predicting the 9 permission questions from the all sentences and not just from the relevant sentences. And it gave a pretty poor result, and this is why we decided to use this 2-step approach too.
Thank you.
Thank you.
Great. Any more questions?
Do we have time for one more question?
Yeah, just for one more question, yeah.
Okay, so you mentioned about informed consent ontology and how your work will contribute to that.
Well, yes, actually, our collaborators already have developed ontologies for the common rule and also for informed consent. So we were hoping that by developing a method to extract data permissions, we could combine with those ontologies to, I don't know, to help future research on data permission identification, and to help to facilitate the sharing and bio-specimen sharing process.